Radhika, you can start. Wait, just wait for the doctor to come back.
Yeah, sure. Yeah, so good evening, everyone, and greetings for the talk organized by Inventa Science Magazine. And we are all excited to host you for this very special talk owing to the very special occasion. In our first edition of the Inventa Science Magazine, which was released on the National Science Day of 2021, we explored the impacts of the last year that lie at the intersection of science and society, the facets embracing the pandemic, and the progression and advancement in the elite arenas. The COVID-19 pandemic may have struck the world, have penetrated through the months in the sectors of education, academia, and recreation, and through different sections of the Indian and international society, often redefining our research relationships and methods. Inventa seeks to explore the same, and this, in this talk, we explore the world of COVID vaccine. Today, we have Dr. Jithesh, an expert in the field of public health. Dr. Jithesh received his MBBS from Calicut University in 1996 and went on to receive his master's in public health from Sri Kshetra Tirunal Institute of Medical Sciences and Technology at Thiruvananthapuram. He is currently an assistant director at the Department of Health in Kerala, and his vast experience over the 20 years in the Kerala government health services have helped the state fight off various infectious emergencies that came its way. Without further ado, I would like to welcome our guest speaker. Welcome, sir.
Yeah, am I audible?
Yes.
Yeah, you're audible.
Yeah, okay. So a big thanks to the organizers, the Inventa Science Magazine and the team behind it, because I always love talking to students, especially done repeatedly, so that it is the people like you, the youngsters like you, who should take the mantle of science and scientific number forward. This is in your hands to resist the concerted effort from various corners to push our nation back to maybe prehistoric times. So thanks again for the opportunity. So I'll be starting with this very simple question of whether COVID vaccine is essential at all. There are so many, lots of diseases affecting the humankind, but the body, it is, if it is in small numbers or if it's not very virulent, it is the innate immunity of the body destroys those microbes. But when the numbers are high or the virulence is so high that the innate immunity can not handle it, then the acquired immunity is called into position. So what happens in acquired immunity? When a microbe enters the body, which surpasses the innate immunity, two sorts of cells are pushed into action. One is the T lymphocytes and the B lymphocytes. These are things that you have learned. I'm just reminding you. T lymphocytes and the B lymphocytes. The T lymphocytes, upon an attack by a microbe, the T lymphocytes differentiate into two major types of cells, the killer cells and the helper cells. The helper cells, B lymphocytes, are a different type of lymphocytes. The helper cells do two things. One is like it helps the killer cells go better and help in killing the microbes. It also motivates or influences, stimulates the B lymphocytes to produce the surface of the microbe and mark them for killing. That is, it doesn't directly kill the bacteria or the virus, but it marks them by attaching to the surface proteins. It marks the antibody. For example, think of a large gathering where somebody has decided to engage other people to kill somebody. And if the hired goons do not know the person, then easy method would be passed by that person, and without his knowing, Mark's system is bucked up for action. The immune system is given the message that you should be ready for action. The action should be initiated immediately. But in case of COVID, the cytokines, which are actually messengers to the immune system to improve the performance of the immune system, it goes away, and then there is something called a cytokine storm. So it is the immune mechanism of the body itself that kills the person, kills the cells, kills the body. So in COVID, there is a special case where the cytokines go run haywire, and then they, instead of their original purpose of protecting the body, the cytokine storm becomes a reason for destruction of the body cells. That is why when people start talking about different methods of increasing immunity, at least in the context of COVID, it is actually, if at all there is something that can increase immunity, your increasing immunity might act as a curse for you rather than a boon. So that is very important in the case of COVID-19. Now coming to vaccinology. Vaccinology is all about, what I keep saying is vaccinology is all about mimicry and trickery. You trick the body, you mimic something, you send something into the body, mimicking something else, and then you trick the body into thinking that the body is being invaded. Think of the like egg of the cuckoo in the crow's nest. The crow doesn't know that it is somebody else's egg and takes care of it very well as it takes care of its own eggs. The crow is not able to count it. The crow is not able to understand the difference. So naturally, it takes care of the cuckoo's egg too. Similarly, when something enters the body mimicking a microbe, the body understands that there is a microbe inside, and then it creates all the mechanisms, including T lymphocytes, the killer cells, the antibodies, everything to attack and kill the microbes. So this is how you trick the body into developing its own immune system against any particular virus or bacteria or any other microbe. So this acquired immunity, the difference between innate immunity and acquired immunity is that innate immunity is a generalized immunity. Acquired immunity is specific for each pathogen. So once you are infected by a pathogen, or it's a fake pathogen, that is what vaccines are. Once it is infected by an original pathogen or a fake pathogen, the body mechanism is activated, immune mechanism activated for that particular pathogen. So next time when that pathogen comes, the body already knows that there is this pathogen I have already recognized. I have already my defense mechanism in store. I just need to activate it. So very fast the immune mechanism gets activated. That is why vaccines are being used to produce immunity against particular diseases. So when it comes to vaccines producing acquired immunity, you decide to try making vaccines. So vaccines mimic the original infection and produce immunity, strengthen the immunity against that particular pathogen. So we'll go in more into details of that. Basically, vaccines are produced by three different approaches. One is the whole microbe approach, the microbe or the organism which causes the disease. The whole microbe approach, or a subunit approach where a part of the microbe is used, or the genetic approach where the genetic material, we'll go into more details of it, where the genetic material of the microbe is used. Microbe is the organism disease. Microbes are several organisms, but here in my talk, I'll be talking about microbes only those that produce disease in humans. So when I say microbe, understand that it is some organism, microorganism, which is capable of producing a disease in humans. Okay, so the whole microbe approach, there are three methods by which the whole microbe approach is used to develop vaccines. One is inactivated vaccines, the second one is live attenuated vaccines, and the third one is viral vector vaccines. This viral vector vaccines is very important in the context of COVID-19. So we'll come to that. Now first we'll talk about the general vaccine principles. So for the whole microbe approach, we'll go into all the three. First one is the inactivated vaccine. Some examples are the influenza vaccines and the IPV, inactivated polio vaccine, which is an injectable polio vaccine, which is being used, developed by Salk, not the oral one. The oral one is different. The inactivated vaccine is an injectable one. So these are the vaccines which are inactivated vaccines. What is done is the bacteria or the virus which causes the disease is killed or inactivated by several methods. It could be radiation, it could be heat, it could be chemicals. By several methods, the virus is killed or inactivated, and that killed virus is injected into the body or introduced into the body by some means. Could be oral, could be injectable, or it could be a nasal spray. There are different methods by which vaccines are introduced into the body. So this killed virus is introduced in the body. So the virus is killed, but the virus has its, except for life, it has all the other things with it. So the body recognizes the surface proteins of the killed virus and is being tricked into believing that a live virus has entered my body. So the body immune mechanism starts developing the T lymphocytes and B lymphocytes against this particular virus, whichever virus is introduced into that body. So a virus, antibody and the T lymphocytes, the killer cells and the helper cells against this particular virus or bacteria, this particular microbe is produced. So the vaccine does not produce any disease, but it increases the immunity in the body, increases the immunity against that particular disease. So when the actual pathogen somehow gets introduced in the body at a later stage, the body is already prepared. Hey, this guy, I know him earlier itself. I've already prepared my defenses against him. I just have to switch it on. Immediately it is switched on and the pathogens are killed. So that is how the inactivated or killed vaccines work. The second whole microbe approach vaccine would be the live attenuated one. It is live. It is also at the same time it is attenuated. Attenuated is like somebody rushing at you with a big knife aiming at your chest. Somebody in between catches hold of that person, removes the knife from his hand and then lets him go. He's still alive, he's still running at you, but he doesn't have that potential to kill you by stabbing you directly into your heart. The mechanism by which he's capable of killing you has been removed from his hand. The same thing happens in live attenuated virus. The virus is live, but it has lost it by several mechanisms. Its capability to produce disease has been removed from the virus. So when that virus enters the human body through a vaccine, again the same mechanism is put into action. The body recognizes that as a pathogen, a disease-causing organism, and then develops immunity against it. So that next time when this same pathogen, the original pathogen comes in, it is attacked by the already produced same factor. That is the identity card of the SARS-CoV-2, the spike protein. So the spike protein in the whole microbe approach, there is a virus, another virus which is not harmful to the human is selected. It could be different types of viruses which are not harmful to the body of the human. The genetic material, the gene which produces the spike protein in the SARS-CoV-2 is detached from the genetic material of the COVID virus and attached to the, you can see it in the picture there in the lower part, it is attached to the genome of the vector virus, the virus which is not harmful. The harmless virus, the genetic material gets added on to the genetic material of the harmless virus. And that virus is then, the vector virus is then multiplied in the laboratory, converted into vaccine. Impurities are removed, storage is properly maintained, and then it is converted into vaccines which can be administered to humans. So these are the three types of whole microbes which are entered into the body. One is the killed virus, second one is live but inactivated, third one is the genetic material is removed and attached to another live virus which cannot cause. So these are three methods by which the whole microbe is introduced into the body without producing harm to the body and at the same time helping the body to gain immunity against that particular disease. Okay. If that is over, then we'll go into the next part, next approach, that is a subunit approach. For diseases like pertussis, diphtheria, meningococcal meningitis, etc., these are vaccines that are given in the regular immunization system in our country, part of the universal immunization program in our country. In that, these vaccines are produced by the subunit approach, where a particular subunit of the virus or the bacteria, like the protein or a part of the capsule, which is the, like I said earlier, which is the identifying part of that particular pathogen. That is the identity card. The identity card only is removed from the pocket of the pathogen, and that identity card is given to the body as a vaccine so that the body recognizes this person and develops its defense mechanism against this person. So that when the original pathogen comes, the body is already prepared. That is the subunit approach. And the last approach is the genetic approach, where you have learned about DNA, the RNA, and especially the mRNA. mRNA, the full form is messenger RNA. It is a conveyor of messages. So the mRNA, which conveys message to the body to produce something like the spike protein in coronavirus, is collected, and then that is introduced into the human body. And there we'll talk about it a little more in detail when we talk about COVID vaccines, which are mRNA vaccines. So that mRNA is introduced by a different mechanism into the human body, and that mRNA gives messages to the body to produce that particular protein or identifying factor, the identity card of the pathogen inside our body itself. So the body itself produces that identifying factor, and then the body itself recognizes that, hey, this guy is likely to create trouble. He was supposed to be outside my body. It was not supposed to be recognized as a foreign body, and the body's immune mechanism develops specific immunity against that particular organism, that particular pathogen from which this mRNA was derived. So that is the third mechanism by which vaccines are developed. So there are three approaches: the genetic approach, the subunit approach, and the whole vaccine approach. Inside the whole vaccine approach, there are three methods. One is the killed vaccine, second is the inactivated vaccine, a live attenuated vaccine, and the third one is the live vector vaccine. So these are the approaches by which vaccines are made. Now we'll go into the details of COVID vaccines that are available. Like I said, the inactivated vaccines, we had 5 mechanisms. Over 3 approaches, we had 5 mechanisms, but only 4 are being used at present in COVID-19 vaccines. Inactivated viruses are Bharat Biotech vaccine, which is indigenously developed in India, named Covaxin. Sinopharm and Sinovac are 2 Chinese companies that are producing inactivated vaccines. Inactivated vaccine, we'll go into a little bit more detail of specific vaccines in the next part of the presentation. Subunit vaccines, which are being developed, is Novavax in USA, which is developed from the protein. It is a protein subunit which is introduced into the human body. It is awaiting final approval. The different phases of the trial are completed, and it is awaiting FDA approval now. The third mechanism, the viral vector vaccines, the Oxford University vaccine, which was then given to AstraZeneca for production. The same thing is produced by the Indian company Serum Institute of India by name of Covishield, which is available. Covishield and Covaxin are the 2 brands that are available in India now, used in India now. Sputnik 5 or Sputnik V, I don't know. Which is now licensed for use in India also, and Dr. Reddy's lab has been selected for that. They have been given the permission to produce it, and they have started production, and they have just launched it for use by the public. Johnson & Johnson has also developed a viral vector vaccine, which is not yet available in India. The mRNA vaccine, the newest technology, there are 2 vaccines presently licensed. Pfizer USA and Moderna USA have developed 2 mRNA vaccines. We'll come to why it is not feasible for us to use such vaccines in India in the next part of the presentation. We'll look at each of it. The inactivated virus vaccines, Bharat Biotech and Sinopharm and Sinovac, the 2 Chinese vaccines. It is the simplest of technology, so the cost is very less. But generally, inactivated virus vaccines are considered to have a slightly lower efficacy than other types of vaccines. But in case of COVID, we have seen comparable efficacy of Covaxin with Covishield. So particularly in case of COVID vaccine, efficiency is not much of an issue when you're using inactivated virus vaccine. So biotech vaccine in India, the Covaxin is an inactivated virus vaccine. The subunit vaccine, Novavax, which I said in USA is awaiting approval. That the protein, the spike protein, please listen to this slide and the next slide very carefully, because there is likely to be a confusion there. The subunit of the protein from the SARS-CoV-2, the spike protein, which I said is the identifying factor in the protein. So the subunit spike protein is produced by a particular gene. The message is sent by a particular gene. That gene is removed from the SARS-CoV-2 SARS-CoV-2 virus and introduced into something called a baculovirus, which is the virus which infects moths. It does not cause disease in humans. So, baculovirus genetic material is the genetic material from the SARS-CoV-2 is introduced into the genetic material of the baculovirus. Then baculovirus, when it starts multiplying, it also produces spike protein of SARS-CoV-2. Baculovirus is a totally different virus, but due to our genetic engineering, we have introduced the spike protein gene into it, spike protein of SARS-CoV's gene into it, and then while it is multiplying itself, it also produces a lot of SARS-CoV-2 spike protein. That spike protein is removed from the baculovirus culture and developed into a vaccine and injected into the human body, so that human body starts developing vaccine like immunity against this particular spike protein, which means it is immunity against SARS-CoV-2 virus or the COVID virus. So carefully note that here it is not the baculovirus that is introduced. Baculovirus produces spike protein which is separated out and introduced in the human body as a vaccine. Whereas in vector vaccines, what is introduced? The whole virus itself is introduced. That is the difference between subunit protein vaccine, Novavax, and the viral vector vaccine, Covishield, in case of COVID. You should note the subtle difference between the two. Baculovirus is not introduced into the human body. The baculovirus, due to the genetic engineering that we have done on its genetic material, starts producing spike protein of coronavirus within its body. That spike protein is removed and cultured into, developed into a vaccine for COVID virus or SARS-CoV-2. So that is subunit vaccine. And the viral vector vaccine, Covishield or the AstraZeneca vaccine, is developed in chimpanzee adenovirus. Chimpanzee adenovirus causes something like our common cold, human common cold, something like that. It causes in chimpanzees. So that adenovirus called CHAD-OX1, that particular adenovirus is cultured and produced in the laboratory. Its genetic material, into recombination technology, is introduced the genetic material of SARS-CoV-2 spike protein. Again, the same thing is introduced here through genetic engineering. And then the adenovirus, chimpanzee adenovirus, genetic material is contaminated by genetic virus, adenovirus, chimpanzee adenovirus, genetic material is contaminated by genetic material from the vaccines. Have been various treatments for cancer, etc., as well as vaccines have been under trial using this technology, but it was not successful so far. But when it came to COVID, the urgent need for development of a vaccine resulted in a series of experiments on using this technology, and finally it became successful. So COVID vaccine becomes the first vaccine in the world to use the mRNA technology. Here the messenger RNA is introduced into the human body. The messenger RNA carries a message to the human body, to whichever cell it reaches. The message is very clear. Hey guys, start producing spike protein. Which spike protein? The SARS-CoV-2 spike protein. So our body also gets that message. Our body starts producing this spike protein, and immediately the immune system of the body recognizes that it doesn't recognize that our own body is producing it, but it recognizes that some outsider has entered the human body. So this spike protein is considered as an intruder, and that is dealt with by the immune system, so that when the original spike protein comes through the original COVID virus, the body is prepared to defend itself from that pathogen. So this is how mRNA vaccines work. It is a Pfizer vaccine. The drawback of this vaccine is that it should be stored at minus 80 degrees centigrade, and the Moderna vaccine at minus 20 degrees centigrade. Think of how cold it is, because our deep freezers, that we store vaccines in ice-lined refrigerators, and for transportation we use deep freezers. Deep freezers, the temperature is only minus 18 degrees centigrade. Whereas for Moderna vaccine, the temperature should be minus 20 degrees, and for Pfizer vaccine, it should be minus 80 degrees. We do not have, even in India, we do not have mechanisms for storage of this vaccine. So imagine the plight of other poorer countries, like countries in Africa. So it is very difficult for such countries to use such vaccines. Also, it is very costly. But these two are the ones so far available that has the highest efficacy rates. It has 95%. It's said to have 95% efficacy. So it is the most efficient of the vaccines, but the high cost as well as the mechanisms for storage, ability to store is lacking in majority of the countries. So it is out of the question for majority of the developing countries. Now we'll come to the next part of the presentation, which is the how a vaccine comes through all these stages of trials and reaches the production stage or introduction into the community stage. Before I go into the stages, there is something called as translation gap. The stages or the phases of research I am going to talk about is the same for both drugs as modern medicine drugs as well as vaccines. I specifically said modern medicine drugs because modern medicine drugs are the only drugs that go through all these processes. Drugs in Ayurveda or Homeo or Siddha or other streams of medicines do not have to go through all these phases, and hence there are other problems related to that. So modern medicine drugs as well as vaccines go through all these phases, but the regulations are more stringent in the case of vaccines than for drugs. It's already stringent for drugs, but it's even more stringent for vaccines because vaccines are introduced into the body of a healthy individual, whereas drugs are used for treating sick individuals. So we should be extra careful when we are dealing with vaccines. So the conditions for vaccines are very stringent. Just look at it. Look at this slide from right to left. For producing one vaccine or one drug, you need to start at the extreme left with 24.3. There's around 24 to 25 drugs or vaccines are thought of. Somebody thinks of many different types of vaccines. Out of 25 which are discussed and thought of and tried to be produced, only around 12 to 13 reach the preclinical stage. That is in the laboratory, where it's produced in the laboratory and tested in the laboratory. Out of 25, only around 12 to 13 reaches the laboratory. And from the laboratory, again, there is a dropout. When it is introduced in the humans in the first phase study, only 8 to 9 reach that stage. Out of that 8 to 9, only 4 or 5 reach the second phase, and hardly 2 reaches the third phase. And then out of that, only 1 reaches the submission approval stage and is approved for use in humans. So this is the amount of effort that goes into production of one single drug or single vaccine. So we'll go into the phases in detail. The preclinical phase or phase 0, then phase 1, phase 2, and phase 3, following which the drugs are approved, the vaccines are approved, and then even after approval. That is the crux of the strength of the modern medicine stream of treatment, because even after that is the strength of science also. Even after approval and introduction into the community, it is the surveillance is continued, and even later the drug might be withdrawn. So people who are very fond of ridiculing science keeps ridiculing us, saying that you people keep on changing your attitudes. But at the same, you say something is good now, and later after 10 years you withdraw that drug, saying that it is not useful. It's not like that. It is withdrawn. Most often it is withdrawn. Maybe some very few drugs are withdrawn because of side effects or long-term side effects, which are not apparent in the initial phases of the study, because initial phases of the study do not take 10 or 20 years. So the long-term follow-up might be done only in the phase 4. So there are some drugs which are withdrawn later like that, but majority of the drugs are withdrawn because we develop better options. So we keep changing our drugs. We keep changing. Science keeps changing. That is why science is the most potent tool. Saying that for the last 5,000 years we have not changed our treatment is not good when it comes to health. It is always good to keep on revising your options and keep on going for better and better options. So one option which was good as of now, when you get a better option, you discard the old option and go for the new option. So drugs are also tested that way. When 2 options are there, a standard option which is available now, a new one is compared against that. Only if the new one has better safety potential or better efficacy, then the new one is allowed to be introduced in the community. So we'll go by the phases in detail a little bit. Phase 0 is the preclinical stage where it is tested in laboratories. In the laboratory, the testing is mainly for understanding the pharmacokinetics of the drug or the vaccine. Pharmacokinetics is like this, the same kinetics as you say in physics. It is a movement. It is a movement of the drug within the body. How is the drug introduced in the body? How it is absorbed by the body? How it is taken up by the body cells? What reactions happen inside the body? And how it is excreted. This is the pharmacokinetics. So in the laboratory study, the first most important factor that is studied is pharmacokinetics of the drug. And then it is tested in, these tests are done in animals, mainly mice or pigs or guinea pigs or monkeys. So it is tested in the laboratory, and once it is successfully approved, it becomes successful and gets approved, then it passes through the next phase, which is the human first human use stage, which is the phase 1 of the clinical trial. It is done usually in 20 to 100 healthy volunteers. Here it should be noted that even if the vaccine is for children, it is only tested in adult healthy volunteers in phase 1. It is not tested in children. In phase 1, because we are studying mainly the safety of the drug along with its side effects and also the dosage. So since we are studying the safety, especially the side effects and adverse effects of the drug, it is not tested in children. It is tested only in healthy adult volunteers. Close monitoring of the health status. We do not know much about the drug. We just know what it does in animals. So it is very, very, very carefully to be monitored. So it is done only in large hospitals where every facility is available, and the health status of the volunteers are kept under very close observation. The immune response of the human body, the response is studied in the animals in phase 0 or the preclinical phase. In phase 1, the immune response inside the human body is studied particularly for vaccines. Once this stage is over and it is successful, only if it's successful, 10 vaccines going into phase 1, only hardly 1 or 2 pass through to the next phase, that is phase 2. In phase 2, the number of volunteers are increased. It is 100 to 300 volunteers, maybe up to 500, or depends on the vaccine and depends on the speed at which you want the vaccine to be studied. But here the difference is that the volunteers are from the age group for which the vaccine is intended to be given. So if it is for children, here in phase 2, the volunteers would be from that particular age group. So in case of COVID vaccine, under 18 years, administration is not permitted as of now, because we have still not studied the efficacy of the vaccine in children up to the age of 18 years. So that study has started now. Once that results are available, we'll introduce the vaccine to under 18 years also. At present, it is only above 18 years. So it is introduced in the volunteers are from that particular age group. And here, either a single blind study or a double blind randomized control trial. Randomized control trial is the gold standard. Some of you might be already be knowing what a randomized control trial is. Randomized control trial is considered to be the gold standard of evidence in scientific literature, in quantitative studies. So what is done in randomized control trial? Something like a computer program randomly allocates the volunteers into 2 groups. So if it is double blind, the person who is administering the vaccine or the person who is receiving the vaccine, both do not know which is which, whether I am receiving a vaccine or a placebo. So vaccine is given to the intervention group, placebo is given to the control group. That is a comparison group. So vaccine and placebo are exactly similar in terms of sight, taste, smell, everything. Both are exactly similar, so that nobody can understand which is vaccine, which is placebo. Because if I know that I am getting a vaccine, my reporting of symptoms, my reporting of effects within me by the vaccine would be influenced by my thought about the vaccine. If I am afraid that there are side effects for the vaccine, I would report more of side effects. So in order that the mindset of the investigator or the mindset of the respondent should not influence the outcome of the vaccine, so it is done in double blind phase 2 trial can be done as single blind or double blind. Single blind is where the investigator knows which is vaccine and which is placebo, but the person receiving the vaccine does not know whether I have received the placebo or the vaccine. He just reports what he is asked to report, and then the investigator finds out whether there is any difference between the intervention group and the control group. So we compare the side effects as well as the immune response within the body, the antibody response within the body. Antibody is tested through blood tests, etc. And then it is compared between the intervention group and control group. If it is significantly better in the intervention group, then phase 2 is considered to be successful. And then it moves to the phase 3 trial, where hundreds of healthy volunteers are used. And it is always a double-blind RCT in case of vaccines. The efficacy as well is compared by comparing the infection rates in those who have received vaccine and those who have not received the vaccines. The intervention group, out of 500 people who had received the vaccine, how many people have contracted the disease? And out of 500 who had received the placebo, how many have contracted the disease in that group? So this study is always done. The phase 3 trial is always done only in places where the disease is widely present. Otherwise, there is no purpose in doing the study because you will not get a result. So this study, phase 3 study, is always done in places where the disease is very commonly seen, so that people have chances of getting infected. You cannot introduce the virus into a human being and see whether he gets infected or not, because that is unethical, highly unethical. So you just observe whether they get naturally infected and compare the natural infection rates between the two groups. So that is how we finish the phase 3 trials, and then it goes for approval. In India, it is the DCGI. In US, it is the FDA, which approves the drugs. For approval, all 3 phases needs to be completed. The results needs to be published, and the whole data should be disclosed to the approval authority, and then finally they approve it. And after that, it goes into the market, and then it is followed up with post-marketing surveillance. In the case of vaccines, there is a reporting system called Adverse Events Following Vaccination, AEFI reporting system. The definition of AEFI is very broad. It says that anybody attributing any side effect to vaccine, you should consider it as AEFI and report. You should consider it as due to vaccine and report. Then 100 reports coming in, you study in detail, maybe hardly 5 or 6 would be actually related to vaccine. But to ensure that nothing is missed out, the AEFI reporting very specifically says that even unrelated should be reported to the system so that cases get missed out. So that is phase 4 trial, phase 4 of the study, which is called post-marketing surveillance. Now I will end with the politics of vaccine. To go into the politics of vaccine, what is very important is to look at the patent laws. Because all drugs, all vaccines are patented, and patent gives the exclusive rights to that authority to produce that particular vaccine and sell it at the price that they decide, except for very few drugs which have Drug Control Act in position. So except for those drugs, the manufacturer can decide the cost of the drug, the selling price of the drug. So this is where the 2 different types of patents come in. You people might be aware of the TRIPS agreement, the GATT agreement and all, which was maybe some majority of, if you would be very young children or maybe not even born at the time of these agreements, but it is affecting our life even now. It is going to affect the lives of our children, our grandchildren. Because earlier we had product patents. Process patents. Process patent means a process is patented. If you have a particular process of producing chicken fry, only you can produce the chicken fry through that particular process. Your process might be like you take chicken and apply all the masalas and keep it in your refrigerator overnight and then fry it the next day. Another person can come up with another idea saying that I will fry the chicken immediately. I will just apply the masalas and then immediately fry. That is a different process. Those 2 processes are different. The second person also has the authority to make that particular chicken fry. That is called process patent. But when it comes to product patent, if I have the authority, I have the patent for a particular type of chicken, recipe. Maybe I have the authority, I have the patent for chicken Manchurian. Then nobody else, no other hotelier, no other chef has the right to prepare that particular form of chicken and sell it to others. That is the difference between product patent and process patent. India, as well as many of the countries worldwide, had only process patents. They accepted only process patents, so that when somebody finds out a new drug, Indians are experts in that. Indians are our tendency to copy had been used very well in this particular scenario, because anybody produces a sort of new drug or vaccine, we somehow find out a way out. We find out a different process by which the same vaccine can be produced. Somebody produces paracetamol through one particular process. We produce 5 or 6 or 60 brands of paracetamol through different processes. So that was allowed. But now somebody who finds out a new drug, a drug called X, nobody else can produce drug X through any other process for the next 20 years. So if I get the patent for drug X, I sell it at a price I decide for the next 20 years, after which anybody can produce it, and then only the price drops. Earlier, the prices used to drop in 2 or 3 to 5 years, but now the prices drop only after 20 years. This affects especially treatments like cancer treatment, where a lot of new drugs, new models of treatment are being identified. Those cannot be copied by other companies, other manufacturers, other countries, and therefore we are bound to purchase it at exorbitant cost. This affects treatments like cancer very much. So this is affecting vaccines also to a large extent. So we have to be aware of the two different types of patents and the trouble we have pushed ourselves into by accepting product patent instead of the process patent that we already had. This one example would give you the extent of that trouble that we have pushed ourselves into. The drugs for AIDS, it was initially marketed by US firms which produce those drugs to South Africa and other African countries. As you know, HIV was most rampant in South Africa. So there they needed drugs the most. Lots of people were dying from HIV and AIDS, but drugs cost around $1,000 to $1,500 per day. The cost of treatment was so high. At the same time, in India, another private firm, which is also a corporate firm named Cipla India, they manufactured the same drugs through a different process and offered to sell it at $5 a day. So imagine the difference. From $1,500, the cost is pushed down to $5 a day in a matter of 6 months to 1 year. So in a very short period, we always blame duplicate drugs. We were more experts in making duplicate drugs, not in the sense of quality, but we manufactured it through a different process and sold it at a very low price. So from $1,500, it was brought down to $5, and then later through international consultation, they offered it to sell it at no profit to AIDS-affected countries at $1 a day. So look at the difference from $1,500 a day. Now, if a new drug is brought into the market, the firms in drug companies in America, in USA, can sell it at $1,500 a day for another 20 years. If they start this year till 2041, they can sell it at $1,500 a day or $5,000, whichever they choose to. At the same time, Cipla in India or any other Indian company or any other company in the third world will not be able to produce it for the next 20 years and sell it at $1 a day or $5 a day instead of the $1,500. So that is the plight of the drug scenario that it is ourselves into it. This affects both drugs and vaccines, and that is one reason why we keep telling that vaccines should be given free. There are different reasons why vaccines should be given free. I cite the most important seven most important reasons. Drug for a disease is my individual need, but vaccine for a disease is not only my individual need. It's also the need of the community, so that the progress of the disease in the community is arrested. It is also a constitutional requirement in India to provide health to the community, to the Indian citizens, without any cost. So vaccines should be, as per constitutional agreement also, vaccine should be provided free. There is a large financial contribution in the development of these vaccines from the, especially in case of COVID vaccines, from the government side. So government has the authority to decide that it should be given free. And there is the biggest challenge to rolling out vaccines in the community for successful vaccination programs is the hesitancy to take vaccines in the community. So that can be addressed only if vaccines are provided free. Even then, it is difficult to address. But if it is a costly vaccine, or if it has to be paid for, this vaccine hesitancy will not have any bounds. So to address vaccine hesitancy, also we need to give vaccines free. And then priorities get changed because of profits. For example, in case of Covishield, half of the production should be given to Government of India at a cost of 150 rupees per dose. That is the agreement between the producer and the Government of India. But the Government of India has recently come up with an agreement which says that of the remaining 50%, they can sell it either to the state governments at 400 rupees per dose or to private hospitals at 600 rupees per dose. So if I am a producer, who would I sell it to? Definitely, I would prefer selling it to private hospitals at 600 and making more profit than selling it to state governments who are willing, like in Kerala, they have purchased the vaccines from Serum Institute and is giving to its citizens free of cost by purchasing it at 400 rupees per dose. If a state government is willing to do that, but the seller or the manufacturer decides that my profit is better by selling it to private agencies at 600 a dose, I gain 200 per dose. So that priorities get changed that way, and then it affects the vaccination program. And for any disease control, the vaccination program should be very easy and fast to implement, especially in cases like COVID, which has very high mortality rate and complication rate. So for easier and faster implementation, vaccines should be given free. And finally, it is a human right to get vaccines and any preventive measure against any disease free of cost for any human being. So these are enough and more reasons to give vaccines free of cost. And finally, regarding the politics of vaccine, one important point I would stress before stopping is something called, you all have heard about apartheid. You know what apartheid is. I don't need to discuss it further. There is a sort of apartheid going on in case of vaccines also, especially in case of COVID vaccine, which many of us in public health use the term vaccine apartheid for it. There is a sort of serious discrimination going on. You can look at this chart. Look at countries which are dark green, USA and other countries which are likely to have universal vaccination completed by end of this year. Look at those in red, which are expected to at this particular stage. This graph was made on 22nd of January 2021. At that stage, the data showed that these countries which are marked in red would complete vaccination at this particular pace only by the early 2023. So look at which countries are lagging behind. Almost the worst hit. So this is a form of apartheid which is propagated by the vaccine manufacturers and the sellers of vaccines. So this is one important thing which we should be. Look at the neighbors of India, Pakistan, Afghanistan, and I'm poor in geography, so I don't know what are the other countries on the other side which are marked in red. Anyway, many neighbors of India, including Pakistan, Afghanistan, Bangladesh, Burma, are all in the red zone. They are the last ones to be. India is in the second last zone, and our neighbors are in the last zone where they will be vaccinated completely only by early 2023. Why is this happening? There are several reasons for it. Most important of which is that 13% of the countries, influential countries in the world, have already booked or purchased 50% of the production for this year, for the whole year. Australia and Canada especially have procured or have already booked vaccines doses, which is 5 times their whole population. If they have 1,000 population, they have already booked 5,000 doses. So all these vaccines are likely to go into these countries only. The rate at which they are purchasing is also kept confidential by the companies so that nobody knows what rate they are selling it to. So the more rate they get, the more doses are sent to those countries. Maybe that is pure business operating there. But UNICEF has somehow managed to collect some data about it, and the data is shown in the table below. AstraZeneca vaccine, which was sold at $2 a dose in Europe, it is being the same vaccine which we are talking about purchasing at 150 rupees to 600 rupees. They were sold at $2 in Europe, but it is sold at $4 a dose to South Africa, Uganda, and other African countries. The irony of it is that South Africa and Uganda are the major countries where the vaccine trials for this particular vaccine was done. For vaccine trials, they needed people from these countries. But when it came to giving them vaccine, the approved vaccine, they doubled the cost of the vaccine. Moderna and Pfizer vaccine, which are sold at less than $20 in USA and Europe, are sold at $25 to $37 in other countries. So if the vaccination goes at this pace, the biggest tragedy is that 85 of the developing countries of the around 180 odd countries in the world, 85 of these developing countries, they cannot complete their vaccination before 2023, which means that essentially 2.5 million people in these countries are going to die. These were preventable deaths. These deaths could be prevented with vaccination, and 2.5 million preventable deaths would happen in these 85 countries before the vaccination is completed in 2023. Look at the extent of vaccine development over the very short period for COVID be translated into an equitable distribution, which gives proper health to both types of citizens, both from the developed countries as well as the poor developing countries, especially countries in very backward countries like those in Africa. Thank you. This is all that I think I have gone a little bit overboard than I had decided to. So for the next few minutes, maybe you can go in for, the moderators can select the questions that have come up, and those that are related to, we'll stick to questions about, mainly about COVID vaccines. That's all.
Yeah, sure, sir.
Patient listening, and I'll take questions if there are any.
Yeah. If anyone in the Google Meet platform has any questions, they can unmute themselves and post a question to the speaker.
Hi, sir. So thank you for that talk. It was a really interesting talk, and I particularly like the analogies you made throughout to explain the concepts. So my question is regarding the different strains of the virus that have been found, and how does the type of vaccine, I mean, you've described many different types of vaccines. So how does the type of vaccine in dealing with different types of strains?
Yeah, we are yet to have final data on it. This is a very new disease. The vaccines are even newer. So as far as available information is concerned, most of the vaccines that are available now are effective, if not fully effective, like the earlier strains. They are also effective against the newer strains also, most of the newer strains. So vaccination program should be continued even if the new strains become more commoner, because it gives a sort of protection. We are yet to get final data on what is the sort of protection that newer variants, like the vaccines, give against newer variants. So data is still coming in, and it has to be analyzed. Maybe it will take some more time to give a final opinion. But as of now, we believe that it is effective. From the available data, we understand that it is like reasonably effective against the newer strains also.
Thank you.
Yeah, anyone else with a question?
Hello.
Yeah, go ahead.
Yes, sir. Thank you for the talk very much. I wanted to ask, how safe or unsafe to bring a vaccine into circulation without testing it on the particular demographic first?
Like, do you mean that particular age category or particular country or something like that?
Country.
Country, okay. Yeah, vaccines and drugs are allowed to be introduced into a country only after results of studies in that particular type of population are available, including like age, sex distribution, the ethnicity. All those factors are taken into consideration. Like a drug is allowed to be introduced, a vaccine is allowed to be introduced only after such kinds of studies. But in case of COVID vaccination, like the stringent norms have been relaxed a little bit because of the enormity of the problem created by COVID. So we haven't insisted on full trials after the trials have happened in some other countries. We have gone for like shorter and lesser number of trials in our country before licensing it into. We have gone into details of the studies conducted elsewhere, analyzed everything, plus details of the studies conducted in our country before approving it. But there is a slight, I would say, slightly. When it came to COVID because of the enormity of the seriousness of the problem.
Okay, thank you so much.
Thank you, sir, for the. But I have a question. This is regarding, in some countries they are mixing vaccines to increase their efficiency. So, what's your opinion on that? Like in China, they are mixing Sinopharm and Sinovac to increase its efficacy. So, do you think it will be effective or?
It depends on what vaccines you are combining. So, especially when new strains are coming up, when one particular vaccine is not effective or not fully effective against all the strains, another vaccine which covers that particular area where this vaccine is weak, combination would have a synergistic effect. Like even in drugs, there are synergistic effects. One drug combined with another drug would have 1 + 1 will not be 2 there. 1 + 1 will be something more than 2. It will be 3 or 4 or 5, something more than 2. That is what happens in the case of drugs. Even in case of vaccines also, if one vaccine which provides 75% efficacy is combined with another vaccine which has 75 or 80% efficacy, the total efficacy would be increasing provided these two vaccines cater to different subsets of the virus, different strains of the virus. But there is no purpose in combining two vaccines which act similarly against the same set of subsets of the virus. So different strains, vaccine A also acts against strain A and B. Vaccine B also acts against strain A and B. Then there is no purpose in, especially if the efficacies are the same, there is no purpose in combining them. So it all depends on which vaccines you are trying to combine and what is the efficacy profile of those two vaccines.
Thank you, sir. Sir, another question I have is regarding we're getting information out of second wave, third wave. So it will continue like seasonal influenza. Like we have seen that seasonal influenza
Yeah.
Can COVID-19 be like that?
Yeah, H1N1 was like, what do you say, a glamour person when it came first. Now H1N1 is something like our neighborhood gentleman. So it is there still going around. It was very serious issue initially, but then we got used to it. It got used to us. So there is less of mutations now, and we got used to each other, and then the severity has come down a little bit. But we are still living with H1N1. Similarly, we will be forced to live with COVID virus in the future also. COVID is slightly more dangerous than H1N1, so we are into a more serious note in that case. But still, it is not going to vanish just like that from our country or from the world. So naturally, we'll have to live with it. That is the importance of fast and vaccination to the masses, mass scale vaccinations at a very fast rate. That is the importance of vaccination in case of COVID, because COVID is not going to leave us alone unless we develop immunity ourselves. The only way to develop immunity is by vaccination. So it should be done very fast. Otherwise, it will take a big toll and then still remain with us. That has been the history of most of the epidemics. Like think of the the biggest epidemic so far has been the Spanish flu, which had the second wave was more dangerous than the first wave. Same thing is happening in case of this COVID pandemic also. Pandemics keep repeating itself, and the names will be different, the viruses, the germs will be different, but pandemics keep repeating. And the history of pandemics is almost the same story. It's like what people say about Hindi films. The plot is the same, the triangular love story is the same, but only thing is Shah Rukh Khan is replaced by Salman Khan, and Kareena Kapoor is replaced by Veetilakath Tandon maybe. So that the same thing happens with pandemics.
Thank you, sir, for a wonderful analysis. Thank you.
Yeah, so maybe we can continue with the questions from our YouTube audience. So the first question from Gokula Karthik, it says, what are your thoughts on the origin of the virus?
Yeah, that is one question I'll skip for now, because we do not have, as of now, we do not have any data to say that it was like a lab-produced virus. There are lots of messages floating around, lots of news floating around, but we can, as a scientific community, we need to have proper evidence. We cannot trust anything unless we have proper evidence. As of now, there is no evidence to say that it is definitely due to something like a biowarfare. But there is also a possibility there. We need to leave our options open until either of the two is proved. As of now, we cannot say it is not there or it is there. Both we cannot say.
Yeah, thank you, sir. So the next question from Ira Sippu. Has there been a hesitancy among doctors and other medical professionals in taking the vaccines? Or is there a necessity for taking the vaccines here?
Yeah, there was what I've seen in Kerala. I am not very familiar with the scenario outside, but all over the country. But in many of the states, there has been a serious hesitancy. For example, in Tamil Nadu has a serious hesitancy when compared to Kerala. Usually in all vaccinations, Tamil Nadu and Kerala are at present over the last few years are running almost parallel, or with equal amount of hesitancy or equal amount of acceptance. But in case of COVID, the hesitancy in Tamil Nadu has been more than in Kerala. But even in Kerala, the state with the highest literacy and the largest number of modern medicine doctors and all, but still there was a hesitancy in the initial phases among both the public as well as the medical community, doctors, nurses, all the medical community were very hesitant, especially when there were many controversies regarding the vaccines, especially in terms of licensing of Covaxin, because in a literate community, these issues are discussed in all fora. So there was a hesitancy in the beginning. That hesitancy is there throughout the country, but it is more in like states like Kerala, Tamil Nadu, where people keep discussing such things and the media is very strong. So there is a large amount of hesitancy even earlier, even in the medical community, but then it has changed. Like we, in our health system here in Kerala, we say that initially we were running behind people with the vaccine. When it was given only for healthcare workers and the frontline workers, we were running behind people. And when it was given for people above 65 and people above 45 with comorbidities, we were again running behind those people, asking them to take vaccine. They kept saying, we'll come tomorrow, we are busy today, we are busy this week, we'll see next week. We have to think more about it. We have to look at side effects. People are saying that there are side effects. There were many reasons they cited. Now, when there is a news that there is going to be a vaccine shortage, people are rushing to the vaccination sites. So now that hesitancy has gone back, and whenever anything is in short supply, people have a tendency to rush towards it. Tomorrow you may not get it, then you definitely go and get it today itself. So there is extreme rush in the vaccination sites in many of these states now.
Yeah. So the next question from the same person, Irazibu, it says, is there a necessity for the results of all clinical trials for vaccines to be publicly available, for vaccine and drugs both?
Yeah, there is something called the Clinical Trials Registry of India. Even a very minor clinical trial which does not use any drug. If you are going to do a trial, a clinical trial with patients or with volunteers anywhere in India, you are supposed to register in the Clinical Trials Registry of India. So any person accessing that particular website, that particular registry can know what all trials are happening in India in the field of medicines or drugs and vaccines. And also, as the trial progresses, all the details are uploaded on the website, and then it is available in the public. It is mandatory that these details are made available in the public domain. So maybe there is a delay in, like a slight delay in the documents being available to the registering, the licensing authority and to the public. There is a gap there, but still, finally, it has to be made available on the public domain.
Yeah, thank you, sir. So the next question from Niranjan A. It says, how justified is the distribution of homeopathic medicine, arsenicum album, in many states? Won't there be a serious consequences where proper studies haven't been performed on the drug? Yeah, that is a big debate going on across the country, especially in states like Kerala. The argument is that it's a system which is approved by the government. But when it comes to the question of science, for any drug or any scientific intervention to be approved or for us to say that it is scientific, the first thing is that the first stage of the proof is that there should be a plausibility, right? The argument should be plausible. It should be believable. It should be something like possible. But look at the basic philosophy of homeopathy. It says that homeopathic medicine, it says that the more you dilute, the stronger it becomes, right? So a philosophy which says that as you keep diluting things, it becomes stronger. It is against present scientific thought, or whatever we know about as part of science, whatever we know now is in contradiction to that philosophy. So that is the basic thing about homeopathic medicine. So arsenicum album is no exception. So the more you dilute it, the 30C is like diluted so much that there is no single molecule of arsenicum in that. So then how does it apply? Now they have come up with various arguments. Various people have come up with various arguments about memory of water or nanoparticles, all which have been disproved. So there is no proof that it is effective, and also no studies have been done even after giving it to the population. How many people have a field trial could also have been done. Instead of that, it is being distributed without any trial, and the danger of it is that because it is diluted to such a low level that there is no arsenic involved in it, that doesn't cause any. It's like drinking plain water, so it doesn't cause any harm as such, but it gives a false sense of protection, and then people would not care for taking proper protection, which is like the SMS that we say, social distancing, wearing mask, those things are not given that much of seriousness if I believe that I am protected by the homeopathic medicine arsenicum album. So that indirectly it causes danger in the community. Yeah, so we have one last question from the YouTube audience. It's from Sai Thorve. It says, do we have any data of how safe the vaccine is in kids or infants and about their side effects during the trials? Do you see any hopes of seeing it being added in the national immunization schedule?
It's about the COVID vaccine, right?
Yeah, COVID vaccines.
Yeah, COVID vaccine, I've already talked about it a little bit. Studies have not been done in below 18 years. That is why, at present, in India, it is not licensed below 18 years. Earlier, we started off with healthcare workers, frontline workers, then expanded it to, it was licensed for use by everyone above 18. But because of non-availability of vaccine, the limited availability, we started off step by step. We started with the healthcare workers and frontline workers, then went on to elderly and those with comorbidities. Then we went on to others above 45. Then we added on anybody above 18. for which some states are giving priority to above 18 with different comorbidities, and then they will go into other people without comorbidities. But as of now, it is not licensed for below 18 because studies are yet to happen. Studies have already started, but the results are not yet available, and it is not in this stage where it can be licensed as of now. So the moment studies are like results are out and it is proven that there is no like efficacy may not be an issue now. The issue is about side effects and adverse effects. So if it is comparable with the adult population, then it will be definitely licensed for pretty soon. It will be licensed for young children also. We hope that maybe in the next maybe 3 to 6 months it will be available for children also. We have already the one of the categories which was earlier not allowed is pregnant women. We have already slightly moved into that category. We have started giving like one of the vaccines have already been approved. The other one is like it is approved, but the manufacturer is yet to give out that call in their schedule. So vaccines are being approved for pregnant women and lactating women. Later it will be available also for young children also, and it may be part of the immunization universal immunization program in India if this stays on as an endemic.
So there has been one last question from the YouTube audience, so hope you don't mind. So it says,
it's okay, go ahead.
Yeah. It says, what is your opinion on coronal kits using scientific jargon to sound convincing and make people buy it, even though there are no significant clinical trials backing it?
Which one? Coronal?
The coronal kit.
Yeah. That is, look at pseudoscience. You're all like students of science. So one thing you should, along with learning science, you should also learn the methodology of pseudoscience. Earlier, they used to sell their pseudoscience by saying it is effective, but people have started questioning back. And now they what they do is not only about coronally, any pseudoscience. What they now market their pseudoscience materials by using jargon which they have borrowed from the scientific community uses. Nanotechnology, they use more of nanotechnology than the scientific community uses. We use the term nanotechnology, nanoparticles only where it is needed. They use nanoparticles everywhere. In any talk, they would bring in nanoparticles when they do not have any explanation. I'll give you one example. Like they are selling sort of mattresses which has like magnetic mattresses which can cure many diseases. It's another pseudoscience. But earlier they used to sell it by saying that it is a magnetic mattress which can prevent diseases, which can cure diseases, and people used to buy it. But now people are more aware of things and they start questioning. So then now they come up with explanations using scientific jargon. They say that there is a magnetic field in the like what do you say? On the Earth there is a South Pole and a North Pole. There is a magnetic field on the Earth. So this like there is a magnetic field. We have iron inside of our body. We all have learned that we have hemoglobin, and hemoglobin has iron in it. So they use those scientific terminology into convincing people, general public who doesn't know much details about the scientific terminologies, to convince. They use these jargon to convince them about their pseudoscience. They say that the Earth has a magnetic pole. So magnetic field. So unless you use this particular mattress which prevents that magnetic field from interacting with the magnetic field inside your body through the iron in the hemoglobin, then it can have adverse effects, and this mattress can prevent that. This is how they sell the mattress now. This is what happens in all cases, including Baba Ramdev's coronal. Not only about that, arsenicum album, any pseudoscience drug you see, any pseudoscience material you see, the same is the situation.
Yeah, I had one question. So the initial guidelines for the vaccination procedure said that the second dose need to be taken, so I say 28 days or 48 days from the first dose. But the recent guidelines extended the gap between them and say it should be taken after 12 to 16 weeks. So is that because of the a situation so that everyone will get at least a first dose, or is it considering the efficacy of the vaccine itself?
No, when we introduced the Covishield vaccine initially, it is only about Covishield. It is not about Covaxin. Covaxin is given at 4 weeks interval even now, 28 days interval even now. The interval was increased only for Covishield. So when we introduced Covishield, the studies were available, but studies were being continued. So people were taking vaccine slightly delayed in some cases due to various operational issues, because they forgot about it, because they didn't have time for it. There are many people who took vaccines at longer intervals rather than at 28 days. So most of the people took it at the correct interval. But when, due to several operational issues, many people took vaccines later with a larger interval, it was found that they had better immunity. So, like I said, the phase 4 trial is still, we are still looking at how the vaccine is performing on the field. That is the beauty of science. We are still looking at it. So we found that the scientific community found that those who inadvertently delayed their second dose had better protection. So naturally, studies moved in that direction, and then finally we decided that we'll go for a longer interval. That was when we increased the interval from 28 days to 6 to 8 weeks, and then later to 12 to 18 weeks. That is the science behind it. And the latest recommendation, I'm not very sure whether, I am yet to go into that. I'm not very sure whether it is purely scientific consideration or some practical consideration as a mass from first doses to the community, so that they get some sort of protection. Rather than giving full protection to a few, we give a reasonable amount of protection to majority of the people. So that could be a practical consideration in the latest guideline, but the initial guidelines, the initial extension of the period between 2 doses was purely on scientific merit.
Yeah, thank you, sir. So I hope everyone's questions were answered in the session, and it was a great talk, and we had a lot of insights into the politics and the work of working, how the vaccination works, and many new things in the talk. So as a part of the Inventa team, I would like to thank Dr. Jithesh for giving us this wonderful talk, and I would also like to thank the Inventa team for organizing this, and all the audience who attended it, and also Parth Ankam, who helped us in streaming the talk live. So thank you everyone for attending this talk today, and hope to see you again sometime. Thank you, sir.
Thank you. Thank you.